18 and 19. Australian scientists have grown a miniature mammalian kidney from stem cells taken from adult skin. So, question 18 is basically asking, in order to develop the technique above, it is essential to know how to do what? So, we're looking for what's the most essential component of this process. We know we've got stem cells that have given from adult skin, but we want these stem cells to go into mammalian kidney. So, the big thing here is there's two steps: differentiation and proliferation. Differentiation is when we get these stem cells, we want them to give rise to the desired organ tissue or the tissue that we want. And the big thing here is gene expression, how genes are actually expressed. Because if the factors that are present when you have these stem cells will respond to those particular genes, and therefore we will produce the proteins that are necessary to produce mammalian kidney when we're in those infant stages. So the big thing here, when I really look at it, is the idea of switching on certain genes. Because if we switch on certain genes, those stem cells will actually divide, or they will give rise to the kidney tissue. So C is the answer in this case. We don't actually need to remove a kidney from a developing embryo. By the sheer means we've got adult skin. So that goes away. We don't need to necessarily insert kidney genes into skin cell DNA. We're actually looking in terms of the idea of gene expression here, the proteins that are produced that will push those stem cells to the mammalian kidney. And it's not about amplification using a polymerized chain reaction. That might come down later down the track, but we know that's for different means as well. So C is the most important. When we go to 19, it says, in the future, scientists aim to grow full-size kidneys for transplant in patients with kidney disease using the patient's own skin cells. This would overcome the problem of rejection of the transplanted kidney by the immune system. So essentially what they're looking for is to actually replace these particular kidneys. So rejection of transplanted organs results mainly from what? Okay, so we know we're looking at non-self cells here. Okay, we're looking at the donor organ. Everything revolves around our immune system thinking that that donor organ does not belong. So it's got nothing to do with the patient's immune cells and nothing to do with the patient's immune system, especially by immunosuppressant drug treatment, because that would help the kidney actually survive. The next point here is it's the donor organ we're looking at, but what would actually attack those particular organs? And it's clearly the cytotoxic T cells. The cytotoxic T cells obviously bind and then obviously puncture the cell membranes and obviously lead to the death and program cell death in terms of apoptosis. So we go in terms of B, which is cytotoxic T cells.